All right. Well, welcome to BrainStream. I am Professor Chris Thompson, and I'm a professor at the Virginia Tech in the School of Neuroscience. So I'm a professor of neuroscience, and I'm going to be using these live streams to just chat about various topics related to how the brain works, how the brain functions. I'll probably end up talking about other things as well. I'm interested in other things. Might really, some very trivial things we might even discuss because, got to reach out somehow with the pandemic going on. And then, mostly I'm interested in making sure that people, have an appreciation for the brain and learn about the brain a little bit. And so I've stepped into, I would say, a topic that's quite controversial, at least in the public's mind, not so much in science, but a little bit in science. And that's the nature of how the brain uses its circuits to create the sense of gender identity, to create the sense of sexual orientation, and how that can differ from biological sex. So I already did one live stream on this, and it got a fair amount of views, which was great. It got a largely negative response from the people seeing it, which is because of the Shapiro fanboys coming to watch it. So I'm going to follow a little bit up on where that had left off. The stream had cut out before I could really finish it up. And so there was that. And so the other thing I'm also planning on talking about tonight is the role of how lead has affected human behavior on a population level, which is kind of getting a little bit closer to some of my current research. But I'm going to be mostly talking about some of the societal roles of lead. And we're just going to talk about a little bit of that. And I plan on revisiting that a few times with these brain streams to kind of organize my thoughts on the broader topic. And I have also found that this is a great way for me to research a topic that I'm interested in, and so that I can be better prepared for my class that I'm teaching right now, which is on neuroplasticity. So I'll be talking about neuroplasticity as well. So in fact, that's part of the reason why I've been talking about transgenderism, because my old research before I started working on more neurotoxicology issues and how that affects how hormones shape how the brain grows. My previous work was more on how sex hormones shape brain structure and function, and they can have a huge influence. So that's where a lot of my interest in this comes in. So I know quite a bit about how hormones can shape how brains grow and how they can even change as adults. So, but I didn't know as much about it in humans, right? Because I didn't study this in humans, I studied this in animals. So I've been working on that issue, researching it because I'm going to be talking about it in my class. And so I thought this would be a good opportunity for me to also get some of this information out into the world as well. So tonight I'm going to start off with part 2. So this is part 2, following up on the part 1, which unfortunately abruptly ended. So I am going to be covering some things that I had already talked about in part 1, but I'm going to recover it a little bit with a little bit more focus on the idea of transgender issues. So, but first, of course, we got to define sex. So if you have already watched through part 1 and you'd rather skip ahead, go ahead and I will post a link up here where you can just skip ahead to get to that part where we left off. But for those of you coming and wanting to get a little bit more context of what I had talked about or hadn't seen part 1, here we go. So sex is, there's 2 definitions to sex. First one is either of the 2 main categories, which are male and female, into which humans and most other living things are divided on the basis of their reproductive functions. So there's a lot in this definition. The two main categories, I've highlighted that because, yes, in humans and most animals, not all animals, but most animals, there are going to be two main categories, which are defined as male and female. And there are exceptions, of course, to that rule. But generally how we're going to define those two main categories are going to fall along the basis of their overall reproductive functions. So there is a 5th grade level at which people have learned about these topics, and that's generally going to be true on the broader strokes of things. But you don't learn the details in 5th grade. You start to learn some of the details in high school, but it's really current research and research over the last several decades that have found that a lot of the details on exceptions to the rule are actually quite interesting and quite informative. And so that's part of what I'm going to be talking about, which are those exceptions to the rule. So that's the first definition. Of course, the second definition is sexual activity. That's what sex is. So let me get my pointer up here. Including specifically sexual intercourse. So there is that, of course. That's the other definition of sex. And that's not really what I'm going to be talking about today. I'm going to be talking about this part, the two main categories. So yes, I would agree that in general, for most humans, you're going to find that sex is going to fall into some kind of dichotomy, meaning like two main categories. Most humans are either male or female. But that major definition becomes a little bit more complicated when you start to look at specific features. So there are all kinds of features that are different across the sexes. Facial features there are all kinds of different facial features, for instance, that are quite different between the sexes. And there are certain features that you're going to define more as male, and certain features you're going to define more as female. And so if you look at this face right here, you can see this is a very male-looking face. This face here is a very female-looking face. And that's because we have, dealt with mostly, most of the people we deal with are cisgendered males and females. And they're going to have certain facial features that are going to go along with their presented sex and their gender identity. And so those things will be generally obvious. But the thing is, that isn't true for a lot of people, in fact. Like, there are plenty of people who identify as male. They're cisgendered. They prefer the company of females. So their sexual orientation is heterosexual. But they will have slightly more feminine-like features, even in the face, or say height, for instance. It's generally the case that males are tall, but not all males are tall. There are short males that are even shorter than the average female. A good example of that is Ben Shapiro. But that doesn't mean that he's female. He just happens to be well below the average of even females as far as height is concerned. So he has a feminine-like feature as far as stature goes, but he's clearly male, right? He's made it very clear that he's male. And you're going to find females that'll have certain male-like features. And then you're going to find some people, depending upon whatever specific characteristic or suite of characteristics you're looking at, they're going to be somewhat androgynous. And if you look at this face, you could easily see this face being like, maybe some females look like this and some males could look like this. It really does look like a blending of these features. And so what that means is that sex is generally just going to be a mosaic. So you have all these different features that could be quite male-like in some males, and then they will be female-like in other features in that same individual. Some of these things will be obvious facial features, or say height, or the tone of one's voice. But a lot of these things can also fall along the lines of behaviors. features of intelligence actually tend to not be so different across males and females. But there are certain mental tasks that actually are different, which I will mention in a little bit. But so there's that. So how do we define sex differences? And so I wanted to go over this again because this is really important. Some features are going to be sexually dimorphic. I see that my video has not come up, so we're going to hold off here a second. And let me switch to my program and see what's going on with my display. Why that's not working. So bear with me here. Aha. There I am. Hello. Yeah, so I disappeared for a second. I set all this up so that I can be on here. Might as well be able to see me, right? So, all right, sex differences. So sex differences, we have sexual dimorphic differences. And some of these things are going to be quite different in their appearance, and there's not going to be a whole lot of gray area in between. An example of this is genitalia. So for the vast majority of males, they're going to have a very typical penis, and for the vast majority of females, they're going to have a vagina. So genitalia generally are considered to be sexually dimorphic, and that's one way in which males and females are different. There aren't a whole lot of sexually dimorphic features between men and women. Most features fall into what we would call sex differences. So a sex difference is one in which there is a statistical difference between males and females. So in this case, let's say that this is height. I know that blue over here is male, but we're going to say that this is female, right? So if we went and measured a bunch of women and a bunch of men, you're going to find that the men are taller than the women. And that's going to be true on average, and you will find a statistical difference. So these are all the little dots representing all the men, and here are the dots representing women. Now, the thing is, within any given feature, you're going to find individuals who break the norm, who are not average or surrounding the average, that are in fact quite a bit more like the other gender. So, for instance, you're going to find some women who are quite tall. And in this population in which we measured the height of men and women, this woman was in fact taller than all of the men. So this was a really, really tall female. And then, of course, there are men who are quite short. Say 5'4". That's well below the average of females. So that individual who would be 5'4" might be right here. So that's another thing to take into consideration. Let's see here. Put that up. And I gotta put this up 1 second here. Yeah, bear with me. Ooh. There we are. Great. And then I'm back here. Perfect. Yeah. Oh, I see the video is not up now. All right. So maybe I need to have, oops, OBS up. Ah, that's the problem. I thought I was up, but I didn't hit transition. I didn't transition my OBS. All right, so now I should be up. So let me see here. I'm still getting used to this, so you're gonna have to bear with me. This is not part of my day job. I'm doing this for fun. Spending my Friday nights doing this. All right, so here we are. Now I see myself. Beautiful. All right. So sex differences and sexual dimorphism. And so most things that are different between men and women are going to be on the order of differences, not dimorphism. You're going to find that some women will have certain male-like features and some men will have certain female-like features. This is true for physical differences. This will also be true for a lot of behavioral differences. And so that's an important thing to take into consideration. in general, across the whole population, you will find differences that are going to be consistent with our idea of what is male and female. But again, there are exceptions, and those exceptions exist, and it's important to take that into consideration. Okay, so I want to go over this again because I think that this is really important. So many people have this built-in assumption that the chromosomes are what determine sex and gender, and in general, that is going to be the rule. That in general, if a human male, this is all the chromosomes from one cell of a human male, all laid out. 1 through 22, and then the 23rd pair is the sex chromosomes, and this is an XY with the dainty little y there. This will be a genetic male. And if this male has a functioning SRY, there are some males that don't, but if it does have a functioning SRY, then this male is going to end up with testes in most circumstances, not every single circumstance. So the way that works is that there's a gene called SRY on the Y chromosome that if it is there, it gets turned on and it starts and it gets turned on inside the bipotential gonad. So when you are first conceived and you're growing as an embryo inside the mother's womb, you have little gonads that are neither male nor female. The default setting for all fetuses is to be female. So we have within us the ability to be male or female depending upon a number of events that may occur. Now, if some events don't occur, you will just end up being female. But if you have certain events that do occur, for instance, having SRY being turned on in the testes, then you are likely, but not always, going to end up being male. So the SRY gene gets turned on, and it promotes the expression of testicular-related genes, which all humans have, both males and females. And then it suppresses the expression of ovarian genes, which both males and females have. So this is being turned on, and this is being turned off. And so SRY also promotes the expression of another gene called SOX9, which is another category of gene called a transcription factor, which also affects the expression of other genes, doing the same thing that SRY does, but it's turning on different testicular-related genes and suppressing other ovarian-related genes, which both males and females have. This ends up turning the gonad, the bipotential gonad, into a male gonad, which we call the testes. So SRY promotes testicular formation, and no SRY leads to female gonad development. So this is showing the two different pathways in which this can occur. So the developing fetus will have these little nubbins deep inside the body that are the bipotential gonads. They can turn male or female depending upon whether there are genes expressed or not. So the male will end up with and actually I'm gonna for this I will change that so that we can see me and then transition and minimize. Great. So that we can actually see what's going on here. So I'm hiding myself for now. And I'm sure there are hotkeys for this, but I got to work all that out. So let's see. There I go. I've disappeared. So if we have SRY, so going back to these bipotential gonads here, if you have an XY chromosome arrangement and there's there's an SRY gene and it's being expressed in the bipotential gonads, you will end up with testes. So the bipotential gonad will turn into testes, and the testes in utero will start to produce all kinds of testosterone. If there's no SRY present, so you have XX chromosome arrangement, then the bipotential gonads default to being ovaries. So they switch over to being ovaries. So that's how that works, right? So here we have genetics leading into promoting the development of the gonads that are going to be sexually dimorphic. And then these are going to have an influence over other secondary sex characteristics. So how does that happen? So this is where hormones start to come into play. Genetics still plays a role here, but now we're mostly going to be talking about the hormonal environment, and in particular, how it's going to be impacting the brain. So this is showing early on in birth, and then from birth, we've got all the way up to puberty, and then puberty up into adulthood. Okay, so this is just showing the timeline of developmental ages up to adulthood. And this is showing the gonadal hormone levels. Blue is male and red is female. In utero, as I said, the testes start to produce testosterone. And so those males start to produce a ton of testosterone, which circulates around the body, including getting up into the brain, where that testosterone will be used to masculinize the brain. That story is actually more complex than what I just said, and I'll get to that in just a bit. But females, they would normally be producing estrogens, right? Especially once they reach puberty. But in utero, they don't. So a typical female will have low to no circulating sex steroids. If you were to give this female testosterone during this developmental window, and we will see examples of that, those females end up having certain masculine-like features. And so that's all what happens pre and postnatal development, so perinatal development. And then, and in fact, there's something called a critical window when hormones are going to be playing the greatest role, especially in shaping how the brain works. And it's going to have an impact on all kinds of features of cellular and molecular development within the brain. So that all that reshaping of the brain, making it more male-like, or say in the absence of hormones, making it more female-like, that's going to have a huge influence over whether the hormones are working or not. So let's see, boop, boop, boop. And going back, I'm going to stick my face up here again. Then we have juvenile hiatus, where there are no circulating sex steroids, and they're going to be sex differences in play. So also certain ways that kids draw will be another thing that will be quite different. Yeah, there are all kinds of differences that happen here. Then once kids hit puberty in their early teens, they the testes and the ovaries start to produce their respective hormones. So males start to produce testosterone, which adds all kinds of additional secondary sex characteristics, such as deepening of the voice, growing hair in funny places. And for females, they also will start to grow hair in funny places, but they get breast development and a number of other things. So and then we have more male and female type reproductive like behaviors. And so this is the typical pattern that's going to be happening for most adults, for most humans. You want to keep in mind that this hormonal environment, yes, in most humans, it's going to be dependent on whether you have testes or not. But there are other things that can influence how testosterone is activated, or say estrogens are activated, that are within the environment, that are independent of testosterone, of testes-derived testosterone, that could influence how the brain works. Those are things that I plan on discussing in perhaps future live streams, but I'm not going to get into that today. But there are environmental factors that can influence how testosterone shapes how the brain grows in these early events. And so you could imagine that certain things that aren't so masculinized, let's say testosterone isn't quite as high in a developing fetus as it might be in most males, that male won't be nearly as masculinized, right? There's going to be variability. Not all males are going to have the same exact level of testosterone. In fact, we know it varies quite a bit in utero, just as it varies quite a bit in adulthood. Some males have really high testosterone and some males don't. So this is also true in utero. That's going to have an effect on how that brain grows. It's going to have an effect on all of these features: apoptosis, neurogenesis and gliogenesis, dendritic growth, myelination, synaptogenesis, synaptic pruning. All of these things in certain, especially in sex-sensitive brain areas, are going to be slightly different. And in certain circumstances, they may be so different that it ends up resulting in changing things that generally will fall into the broader categories of being more male-like and female-like, such as gender identity and sexual orientation. But you're going to find those exceptions. Okay. So there are sex differences in the brain. One of the most well-described sex differences in the brain is the sexually dimorphic nucleus of the preoptic area, or the SDN-POA. So males, so this is deep in the brain. This is in the hypothalamus. So the hypothalamus is that brain area that regulates a lot of basic homeostatic functions of the body, such as thirst, hunger, stress, sleep, lactation, a lot of things, a lot of really basic biological functions. Including sexual desire and reproductive behavior. So this area, the sexually dimorphic nucleus of the preoptic area, if you lesion this in both male and female rodents, those animals are very much less likely to be interested in doing sexual reproduction than animals that are not lesioned. And by lesioning, I mean you like basically burn out these areas, right? So the other cool thing about this area is that it is much larger in males than it is in females. So this is a brain slice, okay? For those of you that know anything about brain anatomy, this is the optic chiasm. This is the third ventricle here. So the ventricles are the empty spaces within the brain that's filled with the brain juice called the cerebral spinal fluid. And right next to those areas is the sexually dimorphic nucleus of the preoptic area. And so this, you can see it's much bigger than it is in males than it is in females. The females, it's like this tiny little nubbin thing here. So females have a little SDN-POA and males have a big SDN-POA. So that's cool. There's already this sex difference. But you can see some even more striking differences and changes that happen within the brains of females and males depending upon their hormonal environment. So let's talk about that. So if you have a genetic female, so this is a female mouse, or in this case, these were rats, that are XX chromosome arrangement, and if they are treated with testosterone during development, during that critical window, they will end up having a male-like SDN-POA. So this is a genetic female, but given enough testosterone activation within their brain, they now have a male-like SDN-POA. And so for male rodents that are gonadectomized, which means that you take out the testes at a very, very early age, they end up with an SDN-POA that is much more female-like. You can see it's much more like the female, and it's much smaller than a typical male that would have had that surge in testosterone during development. And so, again, female rats given testosterone will have a masculinized SDN-POA, and then male rats will have, that are gonadectomized during the critical window, will have a female-like SDN-POA. And this critical window, it's basically starts from embryonic day 18. So this would be the 18th day post-conception to postnatal day 10. So it has this perinatal development. Even more interesting is that if you give a female estrogen, so this is giving a female estrogen during development, they end up with a male-like SDN-POA. So you must be thinking to yourself, what the heck? This is a female hormone. If you give it to a female to a female rodent during development, they end up with a male-like SDN-POA, a very big SDN-POA. It turns out that what's happening is that that testosterone, well, hold on, before I get ahead of myself. So the estrogen here is now masculinizing the brain, right? I told you that testosterone is doing it. But what's happening is that estrogen, it turns out that one of the mechanisms by which the SDN-POA is big is that it has fewer dying cells. So in these rodents that are treated with estradiol, they have fewer dying cells. So apoptosis, that's the scientific term for cell death, is much less in female rodents that are treated with estradiol versus female rodents that are treated with a vehicle. And so you can imagine, so there's a lot more neurons here in the SDN-POA. The estradiol protects those neurons so that they don't die. And that in the females that don't have hormone treatment, they actually have much greater level of cell death, and so all those cells die off. So this is the mechanism by which these brain areas become either masculinized or feminized. Masculinized here, feminized here. But it's estrogen that's doing this. So what's going on there? So the interesting thing about the brain is that most of the sexual differentiation of the brain is driven by estradiol. Okay? So it's in fact a female hormone that masculinizes the brain. So sex steroid hormones are really complex. It's beyond the scope of what I really want to talk about here. But one thing that's really cool about this is that testosterone has the ability to remain testosterone. It can also turn into another androgen that is non-aromatizable, is the term that we use for that. Or it can be converted via an enzyme called aromatase that turns it into estradiol. So early on in development, the male testes produce large quantities of testosterone, but in the brain, enzyme aromatase converts testosterone to estradiol. So it's estradiol that is in fact masculinizing the brain. So then the estradiol acts on the estrogen receptors. So it turns out the SDN-POA, it doesn't have androgen receptors. It doesn't have receptors for testosterone. It has estrogen receptors. So in fact, it's estrogen receptors that get activated to induce male-like changes in some brain areas, including the SDN-POA. But it also ends up affecting a bunch of other brain areas. So let's talk a little bit about the impact that this has on behavior, because it has a substantial impact on behavior. So when we are going through development, the normal typical pathway for most males is that they're going to have this testosterone surge. So this is a genetic male. If he has a testosterone surge, when he becomes an adult, he's going to have a certain hormonal profile that will be very typical of males, where luteinizing hormone goes up and down relatively low. Females that do not experience that surge in testosterone, nor a surge in estrogen, their typical development is that they're going to have large pulsatile, periodical pulsatile releases of luteinizing hormone, which ends up stimulating follicular development so that you can so that ovulation can occur. So she's going to be able to generate eggs. So that's how that happens. Now, males, if you castrate them, when they become adults, their luteinizing hormone profile, their hormonal profile as adults, looks very female-like. And females, if they get treated with testosterone, they end up having a hormonal profile that looks very male-like. They don't have a female-like profile, they have a male-like profile. Now, this has really interesting consequences on behavior. So, males, of course, have genetic sex that is XY chromosome arrangement, and the typical path for a male when they are doing reproductive behavior is that they're going to mount a female. Typical females will do what's called lordosis. So they arch their backs and allow access to the male, is how I will say that. So that's the typical pathway that a male will have. And of course, a typical female has XX chromosome arrangement. She typically does not have a surge in testosterone or a surge in estrogen. Some of them do. And in fact, in rodents, there's this really interesting phenomenon of because, you know, with rodents, they will have litters that could be 8 or 10 brothers and sisters. And if a female in the uterus, if she is between two males, she is more likely to express male-like behavior as an adult. Even though she was not specifically treated with testosterone, it just has to do with where she was located in the uterus. And likely what's happening there is that testosterone was spreading from those males through the placenta and concentrating within that female that's stuck in between the two males. And so her level of testosterone probably got high enough to masculinize her in certain ways, and she acts like a male in certain ways. So, but a typical female will show lordosis, but a female that, like that female that was between two males in utero, she ends up, or if a female was treated with testosterone or estrogen, she will end up doing mounting behavior. She will act like a male as an adult. She will want to mount females. So even though this is a genetic female, XX chromosome arrangement, she has ovaries, right? Because she doesn't have an SRY, right? She was just treated with testosterone externally. Or in that uterine case where you have the females that are in between two males in utero, they weren't even treated directly with testosterone. They just happened to be between two males. Those females are also more likely to show mounting behavior and are much more interested in mounting females than they are to do lordosis for males. And then the males that are castrated early on, they're much more likely to show lordosis behavior. So they are going to raise their hindquarters and allow males to mount them. Despite the fact that they have a penis, they do not have a vagina. Despite the fact that they have they did have testes, but they have XY chromosomes throughout their entire body, they're going to end up showing female-like behavior. So, this is pretty critical stuff. Now, the level of hormone that is treated here can have an influence to how much, say, a female will mount, and also will have certain more subtle effects over the degree to which they are able to attract males as adults. They tend to be very unsuccessful as females in reproducing. They also tend to be terrible mothers. So a very low dose of estrogen and testosterone can have a big influence over their behavior and make them less successful in the reproductive arena. So the level of hormone is also really important here as well. All right, so let's step back a little bit and discuss these steps. So I'm trying to simplify it right here. We have XY or XX as the typical pattern for either getting males or females. And this will have an influence on whether we get testes or ovaries. So this is one step in the process. The genetic step. The next step is the development of gonads, which will either result either male or female, typically, not always, as we're going to see. This is going to have an influence over which kinds of hormones are secreted. But this is a step that is very much, tends to be very spectrum-like, where the amount of hormone released from, say, testes in utero varies quite a bit across individuals. It'll also, in the case of rodents, will depend upon whether you have how many males you're surrounded with. And I believe that there's also even evidence of a similar phenomenon happening in humans. And there's definitely this interesting phenomenon of fraternal birth order having an influence over sexual orientation. So the number of brothers that you have, older brothers that you have, and these are genetic brothers, not adopted brothers, that increases your chance of being homosexual. So presumably something that has happened as your older brothers went through the uterus, they ended up changing the mother's, the way the mother recognizes testosterone. It's thought to be, one mechanism is thought to be an immune-related response to that, which is actually pretty cool. Anyway, these things are then going to have a huge influence over the development of male or female-like brains. And there are big differences between males and female brains. I'm sorry, there aren't big differences. There are more subtle differences, but you can find these subtle differences in many, many brain areas. And I have a little summary of what some of those differences are on another slide. So I wanted to talk about some of the more extreme examples in which we can see the influence of the hormonal environment and its impact on overall gender identity. Now, this is a spot where I wanted to show a video from Ben Shapiro. So let's do that. And I need to do this and all right. So that should be up. So this is a separate video on Ben Shapiro. For some reason, he's known as the transgenderism guy in conservative circles in the United States. Jordan Peterson for Canada. Yeah, so he's kind of the go-to guy for this. So let's.
Yeah. So going off that, I guess you were starting to discuss it. I just wanted to know what your opinion was on sort of the implications of this, not only like a culturally significant issue that you've talked about, more invading into more hardcore science institutions, especially considering your wife's a doctor.
Yeah, she is. As you may know, yeah.
Wow. His wife's a doctor. That's a shock. I didn't know that. He only says it 5 times a week.
So I think that I think it's egregious. I think that biology is biology. It's amazing to me that people who proclaim that they are scientists are so willing to overlook basic biology and proclaim that people who are fully biologically male can magically become biologically female. We're not talking.
Right. So he's distilling this down to this delusion that people who are fully biologically male, and as we've learned, sex is a mosaic, and there isn't a single person who is fully biologically male. They're going to be, I mean, what does that even mean? That they are the extreme, like, is Ben fully biologically male? He's not, in fact. His height is lower than the average female, in fact. So he has certain female-like traits. Height is one of them. I would also say that the depth of his voice, he actually has a relatively high register for his voice. So he's not terribly masculinized as far as voice is concerned. Now, he's male. I wouldn't argue with that. He would identify as male, and I would certainly agree with him. He's male. And presumably his doctor wife has determined that as well. But he's not fully biologically male by any stretch. Stretch meaning like height-wise anyway. So let's go on.
Talking about people who are intersex, I always like when people who debate this issue immediately go to the marginal issues where there actual is blurring of the lines. People who are intersex, or they have some genetic condition, they have Klinefelter syndrome or something.
So he's talking about intersex here. And that's actually what I'm going to be discussing. And I actually think, he's trivializing it. He says, for people to discuss intersex or certain genetic conditions that lead to the blending of the genders. These things aren't nearly as rare as Ben Shapiro is actually making it sound. In fact, intersex individual. So I will talk about at the end about like just how common gender issues are and transgenderism is. And it turns out that many of these like intersex or congenital issues actually are relatively common. So Klinefelter syndrome, for instance, these are individuals who are XXY in their chromosome arrangement. So they have 2 X's and 1 Y. In general, they are largely male in their overall appearance. They have a penis, obviously, because they have a Y chromosome. They have testes. But the testes actually tend to be relatively small and dense. They generally have low sperm count. And then oftentimes people with Klinefelter syndrome end up developing breasts post-puberty. And the other thing that's interesting about them is that they tend to have a greater incidence of gender dysphoria. Now, Klinefelter syndrome is not an incredibly rare syndrome. It's like 1 in 10,000 births, I believe. That is rarer than transgenderism, but transgenderism is also quite a rare issue as well, which we'll see the data for that in just a bit.
Right? That's not what we're talking about here. The actual argument of the transgender civil rights movement is that people who are fully male can actually become fully female. That is obvious nonsense.
So there isn't anyone who is fully male wanting to just become fully female. If one's gender identity is one in which they perceive themselves to be more female-like in how they perceive themselves, and almost always, and we will see, there's actually a ton of data on this, that cognitively they tend to be more female-like than male-like. And this is prior to treatment, prior to hormonal treatment. They tend to score more like females than they do males. And we will see the transgender individuals also have certain female-like features in their brain, or, depending upon if they're male to female, or females that are transgender to males, so trans men, they have more masculine-like features of their brain. There are those individuals, just on average, those transgender individuals, they're not fully male or female. The data shows, even in their brain, especially in their brain, but also psychosocially, cognitively, they tend to have features that match more with their gender identified sex. So a trans man is just going to have cognitive and even brain features that match more male-like than your typical cisgendered female. It's just, we'll see some of those data. So the fact, it's basically, if someone is already identifying as a female, if they feel that their identity, their self-identity is female, already they're not fully biologically male. Even if they have XY chromosome arrangement, even if they have a penis, even if they have testes, and even if they have levels of testosterone that match your more typical male. Already, identity, that is one of the things that tends to be quite sexually dimorphic. It is true that the majority of individuals have a gender identity that matches their chromosomal sex, their gonadal sex, and their genitalial sex. It's true. I'm not going to pretend that most people are transgender. Clearly that's not the case. But if someone is already saying that their gender identity is different, that's a clear indication that they are, in fact, different, and that they're not fully biologically male or female.
Nonsense. It's obviously nonsense. And
obviously
again,
nonsense.
If you are the very idea that a doctor is supposed to sit there and profile you based on what you wish you were is so insane and stupid and counterproductive.
We don't have to go on with that. It's ridiculous. So, let's make sure yes, great. So, there we are. All right. And I got to get my pointer up. Thank you for bearing with me. So we're going to just talk about some of these conditions. So these are intersex. And so clearly Ben has already just dismissed these as having any kind of relevance to this issue. I would completely disagree. I think that these more extreme examples are an illustration of just how the system that I described, chromosomes to gonads to hormones to brain development, can go off of the typical trajectory, and that these extreme examples are great examples of that. One of these is called complete androgen insensitivity syndrome. So these are individuals who are genetic males. Take that out. They have an XY chromosome arrangement. Now, these individuals happen to have a mutation in the androgen receptor. Now, this is a form of intersex that's relatively rare. So it's 1 out of every 200,000 births, if I recall this. So this is significantly more rare than Klinefelter syndrome, and also congenital adrenal hyperplasia, which I will be talking about. Nevertheless, these individuals are fascinating because they are male in their genetic sex, and they have an XY chromosome arrangement. Yet, they are phenotypically female and they identify as female. So how does that happen? Well, it's because they have this mutation in the androgen receptor. So they have testes, right? They have an XY chromosome arrangement, so they have a working SRY, and the SRY turns the bipotential gonad into testes, but these testes fail to descend. The testes, they're producing testosterone, but the testosterone does nothing to the body because testosterone has to act on an androgen receptor. It has to, the testosterone is the key. It has to fit into the lock of the androgen receptor. It's going to do that in any cell that is expressing androgen receptor. And then when that happens, then it's going to be inducing all kinds of genetic changes that will masculinize that cell to turn whatever tissue it is into a male-like tissue. This is all happening in utero. So the testes, because they're pumping out testosterone, but the body can't sense it, they are androgen insensitive. Everything else ends up being female-like. So they have a shallow vagina, but they have no ovaries. They also have no uterus. At puberty, if they still have testes, they generally will develop breasts and they will develop hips. These females are reproductively infertile because they have no uterus, right? And they have no ovaries. Interestingly, they have a very low percentage of body fat. They have very little body odor. In fact, these individuals, they often don't really need to shower very often, which is really weird. They usually have really great skin. They can go a week or more without showering, and no one would know the difference because they don't generate oils on their skin and they don't stink. Pretty amazing stuff. So, genetically male, but clearly very female, and interestingly, even more interesting is that that these individuals, they often don't even know that they have this condition until their teenage years. They are so outwardly female that they don't find out that they have this condition until they discover that they're unable to menstruate. So they go to the doctor, the parents take them into the doctor, and they find out that, oh, wow, actually, you have XY chromosome arrangement, you have testes, you have no uterus, you are never having kids, and so genetically you're male, but, there you go, there's your story. So this is clearly a great example of intersex, but it's also an illustration of just how things can go awry, and that there are exceptions to the rule of, oh, XY chromosomes means that you are male and XX chromosomes means that you are female, because clearly in this case, these are individuals who primarily, almost all, identify as female, but they have testes, they have really high levels of testosterone, and they have an XY chromosome arrangement. Okay, so it clearly shows that these things can change. This is an example of someone who had to deal with this. So this is Maria Jose Martinez Patino. She was an Olympic hurdler for Spain. And in the 80s, this is when there were all kinds of controversies around people abusing hormones, like especially the East Germans and the Soviets. They would be using testosterone to boost overall muscle growth. And so in the later part of the 80s and the early 90s, the Olympic committees were doing all kinds of testing to ensure that there wasn't anyone cheating by using testosterone supplements. And this athlete was excluded from the Olympics and excluded from various world championships in the late 80s because when they measured her hormone levels, she had really high levels of testosterone and she had an XY chromosome arrangement. So for that, they're going to exclude her. And that's because at this time, we didn't have a very good understanding of complete androgen insensitivity syndrome. It didn't matter that she had high levels of testosterone. In fact, they could take her blood and just replace it with just liquid testosterone, and it wouldn't make any difference because her body is completely insensitive to androgens. She has no androgen receptor, despite the fact that she's genetically male. Remember, that default pattern of the fetus is going to be towards female. So that's just how that works. All right. So another form of this, another form of intersex that Ben would utterly dismiss as having absolutely no relevance to this issue of gender identity, is called congenital adrenal hyperplasia. So congenital adrenal hyperplasia these are individuals who have a mutation on the 6th chromosome for an enzyme called steroid 21 hydroxylase. So this is one of the enzymes that's critical for making cortisol. So this would normally be expressed in the adrenal glands. The adrenal glands sit on top of the kidney, right? The kidney is the renal system, and the adrenal glands are on top. Adrenal. That's where it gets its name. So in the adrenal glands, you have a certain hormone path that would normally lead to cortisol. But because these individuals have a mutation for CYP21, so they cannot produce cortisol, and instead, because they're not able to produce cortisol, they get buildup of the precursors to cortisol, and this buildup ends up forcing the adrenal glands to start to produce testosterone. So this can happen in males and females, and there are things that happen to the males who have this condition. But the females are particularly interesting. So congenital adrenal hyperplasia in females, what this means is in utero, their adrenal glands are pumping out testosterone. They're making a ton of testosterone, which ends up masculinizing various parts of their body. So these are females. They have XX chromosome arrangement. They have ovaries, right? The ovaries are not producing estrogen because that's what ovaries do not do that in development. But their adrenal glands are producing testosterone, acting like the testes would in a man. So the testosterone then acts all over the body and starts masculinizing certain features. So they tend to have certain problems with menses. They have certain skeletal features that are different. They have what's called virilized female genitalia. So they have an enlarged clitoris. In fact, in many of these individuals, it can look like a micropenis. So basically the clitoris ends up looking like a penis because that's what the penis emerges from in development. The testosterone acts on the preclitoral region and enlarges it and turns it into a penis in genetic males, typical males. And so in these individuals, they're also developing a penis. The vagina starts to close. It's in some individuals, it's entirely closed, and they would have to open it in order to have a more typical like vagina. So yeah, they have all kinds of issues, right? So, and also in their brains. So in their brains, they have differences in the amygdala, which is really important for emotion and fear regulation. They have certain differences in white matter, which we'll talk about, and certain differences in the temporal lobe. So one thing that's really interesting then is about these individuals. So these females, they have been hit with all kinds of testosterone. So this is like that rodent experiment that we already talked about. So this is basically like giving testosterone to females in utero. And so it turns out that these females tend to have a lot more gender and sexual orientation related, I guess, issues or features that are much more masculine-like. So the majority of females they identify that are treated this way, they with congenital adrenal hyperplasia, they identify as female. And of course, that's partly because these females, they can, when a female is born with congenital adrenal hyperplasia, you can see right away. As soon as the baby comes out, you see that it has this misshapen genitalia. So you already know what's going on, that they have congenital adrenal hyperplasia. And so then what you need to do is you start treating those females with cortisol. And then you can start to regulate the testosterone. So then the testosterone won't be produced. So they're not getting the full dose of testosterone that, say, what was happening in the rodent experiments. Nevertheless, around 5% of females with congenital adrenal hyperplasia report gender dysphoria. This is a lot more than what is found in the general population. A lot more. This is like 20 times as much. So they're 20 times more likely to have gender dysphoria issues than the general population. Around 6% have same-sex partners. So despite the fact that many of these individuals would end up saying that they are female, they are lesbians. So now you have another 5.8% that have a sexual orientation that is male-like and that they prefer females. And other studies have shown that somewhere between 20 and 47% of congenital adrenal hyperplasia females have homosexual fantasies. During that juvenile hiatus, when there really isn't any high levels of testosterone at all or estrogens, they have a tendency to show preference for boys' toys and also roughhouse behaviors. So they tend to show male-like behaviors that you would typically associate with males. They tend to act like tomboys. They tend to draw like boys. They also tend to do better on certain cognitive tasks. So one difference between males and females, so a regular cognitive difference, is the mental rotation task. So what this is taking a 3D object, like a picture of a 3D object, and then having it rotated in one direction or the other, like on in a 2D space. Then you ask that individual, well, did it rotate right or did it rotate left? Males do a better job of saying whether it's rotated right or left. So they have this ability to do mental rotations better. And this is one of the relatively few cognitive tasks that is regularly sexually dimorphic sexually different between males and females. An exception, though, is for congenital adrenal hyperplasia women who had circulating levels of testosterone during development. They tend to do much better on the mental rotation test than females so females, the control females. So these are control females. They are scoring like your typical female, and the congenital adrenal hyperplasia test, where they didn't have a mental handicap. So sometimes these individuals end up having mental handicaps, and so obviously they're not going to do very well on the mental rotation test. They do much more. They do much better on the mental rotation test than the control females. So there's that. Another thing about sexual gender identity and orientation is that you can find certain sex differences that are in humans that also appear to seem to coincide with sexual orientation. So humans don't have a very clear SDN-POA. All right, we have bigger brains than rodents, much bigger, and so our hypothalamus is also equally bigger. And so our hypothalamus is also a bit more complex. We have a lot more subregions. There are a bunch of these subregions in the same kind of general area where the SDN-POA is in rodents that are called the interstitial nuclei of the anterior hypothalamus. And we have a bunch of numbers for those. So INAH3 is one of them. And this is one brain area that is larger in men than it is in women. There's also evidence, there's a few studies that seem to show that this area is bigger in heterosexual men than it is in homosexual men. So homosexual men who prefer the company of other men seem to have a more female-like INAH3, and this is actually, the analogy here is to what we had seen with the SDN-POA in gonadectomized rodents. This has led to the idea of a hormonal influence for overall sexual orientation. The majority of women are going to be heterosexual in their orientation, and the majority of men are going to be heterosexual in their orientation. So they're going to prefer the company of women, and heterosexual women will prefer the company of men. In development, heterosexual men are going to have a high level of testosterone that they're exposed to, but it's a range, right? Many of them are going to be much higher than women, but not all of them. There's going to be some who who are going to have relatively low levels of testosterone. And this is just a hypothesis, but the hypothesis here is that these men who fall below, say, some certain threshold for activation of the estrogen receptors in the brain through the aromatization of testosterone to estrogen, that if they're low enough, what you're going to end up seeing is that they're going to be more female-like in their overall sexual orientation, and therefore they're going to be homosexual. And then the hypothesis is that for women who have relatively high activation of these hormone pathways in the brain during development, they're going to be a bit more male-like in their overall orientation, and therefore are going to be homosexual, preferring the company of men or women. So this is a hypothesis that where this feature that we know can vary quite a bit in utero, the amount of hormone exposure, and hormone exposure is not just simply levels of hormones that the fetus is producing. There are a lot of other things that go along with that can influence how much hormone is getting up into the brain and how much it's being affected by, say, the mother's uterine environment. The kind of chemicals that she's exposed to, the mother, can have a huge influence over all of these features and potentially be pushing the fetus in one direction or another to make them more male-like or female-like in their orientation that will then be different than their overall chromosomal sex, different than their genitalial sex, different than their hormonal sex, and different than their gonadal sex. Right? This is all perfectly consistent with all the scientific literature that we know. And, I try to put citations for all these papers. This is all scientific literature. You can go look up everything that I've cited here. Most of this is available for free on the web. You can go ahead. I would encourage you to read as much as you can about this. So, what this means is that getting to orientation and to gender identity involves many, many steps. You have the chromosomes that have to be involved. They have to turn on certain genes. They activate within the bipotential gonads, and then they will influence how those genes work and drive the bipotential gonads to be either testes or ovaries. Those will then end up creating a certain kind of uterine hormonal environment that has to make its way up into the brain. But then in the brain, that hormone needs to be converted to another hormone, testosterone to estrogen, and it then has to act on the brain in order to masculinize or feminize. So there's a lot of steps in here in which pushing in one direction or another could end up with an end result that's going to be deviating from one of, say, the beginning steps, such as chromosomal sex. So it's unfair to say that gender identity and sexual orientation is completely defined by chromosomal sex. It is true for the majority of humans that is the case. This is the typical pathway. But biology is messy. There are going to be exceptions to the rule, and you will find that certain influences that are environmental in nature or genetic in nature, combination of the two, will end up shifting how the brain grows and how it develops and converting it from being, say, more masculine or to more feminine. Remember, the brain kind of has a default the body has a default pattern to be feminine. In the absence of, say, circulating sex steroid hormones in utero, the brain is going to ultimately be feminine. And that means even in genetic males, there's the potential for that brain to end up being feminine, say, for instance, in congenital, incomplete androgen insensitivity syndrome, right? They have a very female-like brain. They identify as female despite the fact they have an XY chromosome arrangement. So something gets messed up here because the brain is not sensitive to testosterone or estrogen in this case. So that is something that's happening, right? The same can be happening for gender identity, and that the shaping of the brain could end up being outside of that range of what we consider to be. So this would be on the tail ends of the distribution. Let me go back. This is a bimodal distribution here. This is a bimodal distribution here. They're going to be different. If we do a statistical measurement of this, you would find a clear statistical difference between men and women in their testosterone concentration or their attraction for women to men. But you're going to find some overlap, right? And that's okay. Those are those exceptions to the rule. They exist, and we have to consider that they actually do exist. This is just for orientation. You could imagine a similar kind of distribution pattern that maybe is, the overall epidemiological data would indicate that there are fewer gender identity. There are fewer transgender individuals than say homosexual individuals. They make up a smaller percentage of the LGBTQ pie. But you still could imagine that on that tail end of the distribution of how gender identity is generated in development, that you could have a brain that ends up being a bit more feminine-like in a genetic male, or a brain that's a little bit more masculine-like in a genetic female. So that's what I wanted to show next. So what are some of these differences that you can see? So this is kind of a really nice summary of this. So in utero, you're going to have in males, they're going to have high levels of testosterone, and it's going to be converted to estrogen to masculinize the fetal brain. Then we see certain differences in the brain, including in adolescence. So in adolescence, we see more between network connectivity in the brain, and in females, it's more within network connectivity. So there are these different networks that we find within the brain of how interconnected it is. You can separate those different networks, and males have more connectivity between those networks, and within females, those networks have more connectivity within. Males have a larger gray matter volume, so that's where you're going to have all the cell bodies. So their cortex tends to be thicker in males. This is something that continues all the way up into adulthood, where males have on average, a larger total brain volume, more gray matter volume. They also have more white matter volume. They obviously have more cerebrospinal fluid since their brains are bigger. They have a higher proportion of white matter, whereas females have a higher proportion of gray matter. So males tend to have a lot more of the connections between neurons. There's other differences. So males have a larger volume of the central division of the bed nucleus of the stria terminalis. They have better visual, spatial, and mathematical ability. And they have weaker right-hand preference. You tend to find more lefties that are male than females. And so females are opposite of all those things. They tend to have higher global cerebral blood flow. They also have better perceptual speed and fine manual dexterity. They also tend to do better on word-related tasks. So big differences in the brain and just overall shape of the brain. Sorry, I shouldn't say big differences. They're relatively small differences, but there are quite a few of them, and it's in many areas of the brain where you find these small differences between male brains and female brains. So what does this look like in transgender individuals? So there are a bunch of studies out there. I'm only going to talk about 2 of them. I encourage you to go and look. Here's a study. Here's the citation. Please go look. So in this study, this is published in the Journal of Neuroscience, a very high quality journal. The title of this is White Matter Microstructure in Transsexuals and Controls Investigated by Diffusion Tensor Imaging. So what they did is they took transsexual individuals. These were either female to male or male to female. So this would be a trans man. This would be a trans woman. They had female controls, that's the FC, and male controls. And they put them into brain scanners, and then they looked at how their brains are shaped. They did all kinds of measurements of overall brain thickness and measuring certain sizes of the brain areas. They also looked at the white matter areas of the brain, and so that's what they're showing here. So this is showing at one part of the brain where you can see the many of the areas of the corpus callosum. So the corpus callosum is a giant bundle of axons that connects the left hemisphere of the brain to the right hemisphere of the brain. It allows the two hemispheres to talk to each other. This tends to be different between males and females. So let's just compare. So the body of the corpus callosum, this is the MD is the diffusion tensor. So it's this feature is greater in the control females than it is in the control males. This is also true in a different area of white matter, where that's called the superior frontal blade, right? So they're looking just on the right side of the brain. So they're looking right over here. And here, the body of the corpus callosum, they're looking over here. So this is the corpus callosum right here. And you can see here, the difference is even greater between the controls that are females and the controls that are males. Just another one of those differences, right? And this is related to that white matter interconnectivity. Now, the interesting thing is for the transsexual individuals. The individuals that are trans men, so these were natal females that identify as males. Their scores for the body of the corpus callosum is more male-like. So this is the females, control females, and you can see that the female to males are more male-like. Now, the control males are actually more female-like. You can see the average is up here. So this part is showing the average. So these are more female-like. And this is even more exaggerated when you're talking about this part of the brain called the superior frontal blade on the right hemisphere. And you can see that the female to male areas are more male-like, and the male to female, so these are trans females, are more female-like. You can see they're right in between. So they're not exactly female, but they're much more female-like than your average male. And the same goes, but in the opposite direction for the trans men. So these individuals have brains that are somewhere in between males and females. That's just this one measurement from this one paper. There are, at this point, several dozen studies just like this. Here's another study. So unfortunately in this study, they have a table that describes all their data because they have too many things that they measured where they found differences. So they had to present it all into a table. This is a study called Cortical Thickness in Untreated Transsexuals. So just like the previous paper, these were individuals who were not treated with hormones. We in fact see transgender individuals, once they get hormone therapy, that the differences tend to get even greater in their brains. The hormones start to act on their brains and they start to turn them either, say, more male-like, or if they get treated with estrogens, more female-like. But this is prior to treatment. This is before any hormone treatment. So the first thing here is to find some female to male differences, right? So here's a bunch of different areas of different parts of the brain, the inferior parietal, the postcentral gyrus, I presume, and this is the Partrian Goliaris. The main thing here is to look at the p-value. So anyone who knows anything about science, and if you've done any statistics, if your p-value is less than 0.05, that's a significant difference. Now, when they also looked at brains of female to male transsexuals, so these are trans men, and they compared them to male controls, they see that these were natal females, but they feel like males, that there were certain differences. And then male to female compared to male controls, also certain differences. Even more so than these individuals. So the brains of these transsexual individuals had thicknesses. So mostly what they were looking at is the thickness of the cortex across many areas of the brain. And they're finding differences of the transsexuals compared to male controls. They have another table where they compare it to female controls, and they also see additional differences. So another thing that they found in this study, this is from the same study, they looked at the volume of the right putamen. So the putamen is a part of the basal ganglia. It's a really important part for overall motor control. There actually are quite a few sex differences that we're now discovering within these areas of the basal ganglia. So this is the putamen is part of the striatum, and there's some really interesting sex differences that are being discovered there, and that hormones can have a huge influence there. In this study, what we're seeing is the size of the right putamen between males and females, and that in males it is significantly larger than it is in females. In females these are trans men, so they are natal females. These individuals are somewhere in between both males and females. So, very interesting, these differences. So to summarize this, I presented the scientific overview, and this is, just kind of scratching the surface of this issue, that the role that chromosomes, the gonads, hormones, all of these things have a huge influence over how the brain grows and develops. The brain is incredibly complex. It's going to be something that is going to be influenced by all kinds of factors, including variation in hormones, the timing in which hormones get up into the brain, environmental factors such as certain environmental contaminants. Many, many features are going to end up influencing how the brain gets shaped. In general, every person would admit, of course, including transgender advocates, clearly the majority of people are going to fall into being cisgendered, and even then, most of those individuals are going to be heterosexual in their orientation. There's no argument there. But it's not just a simple wish that transgender individuals want to switch from being male to female. If they identify as the opposite of their natal sex, that's something that's core to them. It's something that's going to be heavily influenced by the brain. The internal state of what we feel about ourselves is not independent of the brain. It is entirely generated by the brain. The internal sense of who we want to have sex with is not just a choice that exists in the ether. It is something that is heavily influenced by the brain. We know this. This is a fact. There's no argument about these things. There's no other place it could come from. Where else would it come from? Where does gender orientation come from, or gender identity come from, if it doesn't come from the brain? It's deeply rooted in the brain. And we know that there are certain brain areas that seem to be related to these things. So I just think that it's really important to recognize that these exceptions exist. They are important. And, that you got to take these things into account. So I wanted to show one more thing, which is this. Yeah, we'll go here. No, this. So this is from a study. Transgender population size. Oh, actually, I gotta turn off my... Turn this and hit escape because you can't see anything. Okay, so here we are. Transgender population size in the United States, a meta-regression of population-based probability samples. Okay, so this is just looking at the overall proportion of transgender individuals, looking across survey years, and there appears to be some increase in the number of individuals who are transgender. That does seem to be something that's happening. This is probably because over the last decade or so, people who identify as transgender are feeling a little bit more comfortable in talking about being transgender. This is just something that has been so controversial that these individuals just would be hiding the fact that they feel transgender or they identify as trans. So this increase likely is being driven by this. But the important thing here is to look at the proportion. So this is the proportion of the entire population. It's a small percent. No one is pretending that like the majority of people or like the majority of kids are going to end up being, classified as transgender. That just, that we know that that's not the case. But there is a relatively small percent of these people that exist. And it's important that we recognize that they do exist, that they have value as human beings, that they deserve support. And that in my opinion, they really deserve to be treated how they prefer, as far as their orientation is concerned. And we have to accept that, certainly there will be individuals where we won't be able to say, oh, well, you've had this intersex experience, yet somehow you're trans, and obviously you've had transgender issues growing up, and we can connect those dots really easily. There are going to be plenty of examples of individuals at this point who we know that they identify as transgender. We can look in their brains, and they're most likely to have certain differences in their brains, but we don't really know how they got there. It's important to admit that these people exist, even if we don't know all the details of exactly how that happened. And you just got to be open and, open your hearts and love people for who they are. And, that's how I feel about that. So, a few people watching, which is kind of fun. No comments, which is all right. I wanted to go and talk about another thing because what I'm going to do is I'm going to divide this live stream into a couple of different segments. So these will be separate videos. And so let's see here. The next thing I wanted to talk about is about lead and crime. So why don't we go to that? Hopefully that shows up. And I'll get my pointer up. Boom, boom, boom. Laser pointer. One thing that we know about lead. So lead is a metal, right? It's an important industrial component that had been used for centuries, going all the way back to ancient Roman times. They had heavily used lead. And even then, they knew that like getting exposed to lead would have certain consequences. They knew that high exposure to lead would lead to certain forms of cognitive impairment. This is data showing that blood lead levels related to IQ scores. So the average typical IQ is going to be 100. This is the typical IQ score. There are a bunch of studies. This is a summary of various studies that have been summarized in this review here. So this was like a meta-analysis that they had done where they were able to create this curve on overall blood lead levels and its relationship to IQ. And what we can see is that there is a pretty steep decline in IQ at relatively low levels of lead exposure. Many people will say that there is no safe level of lead exposure, and they're probably right. Like, you should definitely not be exposed to any lead whatsoever. Now, there are definitely, though, there's definitely a relationship of more lead being worse. That's certainly the case. So as you increase lead concentration in the blood in children, it's going to be associated with a declining IQ at 40 micrograms per deciliter, which is quite high. That's a 10-point decrease in IQ score. 20 micrograms per deciliter is around a 7-point decrease in IQ score. Okay. So I want you to keep these things in mind as we go forward. So 20 micrograms per deciliter is right here, 7-point decline. This is 10 micrograms per deciliter, around 6 points. And 5 micrograms per deciliter is now considered the level at which is considered to be of concern in children. So if you measure the lead levels in blood of any child within the United States, you're going to detect some, especially if you have sensitive enough instruments. But in general, it's going to actually be quite low. It'll usually be quite a bit lower than 5 micrograms per deciliter for many children nowadays. But you will find areas where it's actually much higher. In fact, one of those areas where this became an issue was in Flint, Michigan. So everyone knows about the Flint, Michigan water crisis. This is Mark Edwards. He's a professor here at Virginia Tech, and he's kind of known as a hero of Flint. He was asked by many community members to come in and test their water. Because in Flint, Michigan, this is the water that was coming out of their taps. So he's got it in the bottle here. You can see that doesn't really look like water. I mean, that's dark orange. It's pretty rusty. And he has in comparison here Detroit water. So what had happened was that in Flint, Michigan, they were having budget issues, and the governor there fired the mayor and fired all of the town council, and he put in some like a CEO representative, essentially. He appointed someone to run Flint and to get their financial situation in order. And so when that happened, they were cutting costs all over the place. And one thing that they decided to do was that they didn't want to buy water from Detroit anymore. So Flint was getting its water from Detroit because Detroit water, they do all kinds of processing of their water, and it's very nice and clean. What they decided to do was turn on their old systems in Flint and just take water out of the Flint River. The Flint River is heavily polluted. It's very corrosive. So it's got a relatively low pH. And what had happened was that in the water treatment plants, because it was so acidic, what they should have done is used what's called corrosive control measures. They should have added certain chemicals that would have changed the pH so that it wouldn't then affect the pipes throughout Flint. Why is that important? Well, Flint has lead pipes throughout their entire water system. This isn't unusual. There are thousands of communities across the United States that have lead pipes. Many of them are not affected by the lead there, and that's because you end up getting this biofilm that collects on the inside of the pipes. So as the water is flowing through the pipes, you get a biofilm that'll collect there, and as long as the pH is relatively high, the biofilm will not, and the scale, there's like a calcium scale that'll build up on the inside of the pipes. It separates the water from the lead. But if you get acidic water in there, that barrier starts to break down, and then the water starts to absorb some of the lead. And this is what was happening in Flint. So this is showing some examples of what this looks like when you measure this. So in water, cause for concern is 5 parts per billion. So if you were to look in some water, if you looked in a cup of water that had 1 billion molecules of water, if there's 5 molecules of lead in there out of the 1 billion molecules of water, we know that this will start to increase blood lead levels. So this is not a lot. This is very little. Detroit water has 2.3 parts per billion. Troy, which is another town in Michigan, had only 1.1 parts per billion. So, and so both of these are below the cause of concern. In Flint, it was 27 parts per billion. So more than 5 times higher than the cause of concern. This was on average out of hundreds of samples that they took. In the Virginia Tech sampling regime, so they were taking hundreds and hundreds of samples. The highest one they found was 158 parts per billion. And there were other samples that were done, one of which found 13,000 parts per billion compared to 5 parts per billion. So this is a major issue. So people are obviously getting exposed to lead. Here's some statistical analysis that was put together by Reuters. So they had this really great study. I should put a link in the description for this part to this Reuters study. So this is showing Flint, Michigan here. Here's the Flint River. And it's showing the percent of children that have tested for elevated blood lead levels. And you can see in this area of Flint, we've got, outside of Flint, it's less than 5% of kids testing for blood lead levels that are greater than 5 micrograms per deciliter, right? In most of Flint, though, it's somewhere between 5 and 10%. And then in the downtown areas of Flint, it got up to about 10 to 15%. So somewhere on the order of 10 to 15% of children were testing for elevated blood lead levels, 5 micrograms per deciliter. So that means, though, that 85% of kids were testing below the level of concern in which we know that there's going to be the increased chance of decline in IQ as well as other behavioral issues. But that, 50%, that's bad. This is the thing, though. Flint actually isn't unique. It turns out that across the United States, you can find children that have elevated blood lead levels in most urban areas. So this is Flint right here. So this is, we're zooming out of this map. And here's Lansing. You can see it's also pretty obvious. Grand Rapids is even worse than Flint. So the children there are testing, there's 15 to 20% of the kids there are testing for elevated blood lead levels. This is also true in parts of Jackson, also in Kalamazoo. And then look at Detroit. So these are children that are testing. So 25 to 30% of kids, upwards of 35% of kids in Detroit that are testing at having blood lead levels that are greater than 5 micrograms per deciliter. So this is Michigan. It turns out that if you look at most urban areas, you're going to find this phenomenon. So this is true in Milwaukee. This is true in Baltimore. You can see in Baltimore, there are areas where it's more than 40% of kids are testing with blood lead levels that are greater than 5 micrograms per deciliter. This is Cleveland. Most areas, are really bad. A lot worse than Flint. So Flint obviously is something to be very concerned about, but it's clearly not restricted to Flint. That this is an issue across the United States, and it tends to be just an urban issue. And then we're talking primarily about kids who live in very poor areas, and we're talking primarily about people of color, so that are going to be affected with this. This is the environment that they're growing up in, and where more than 40% of the kids are still being poisoned with lead. What are the sources of this poisoning? So there are 3 main sources of lead poisoning in the United States. So lead piping obviously is one of them, as we talked about. Paint is another. So paint had been leaded for a very long time. It made the paint sort of go lay down on whatever you're painting. It laid down nicer. The colors were better. There were certain reasons to add the lead into paint. This was generally true in the early part of the 20th century, and lead was started to be taken out of paints, I think, in the 50s and 60s. It might have even been earlier than that. It's the 50s. It was in the late 60s when they started to take lead out of paint. And it was being added in the 19-teens and 1920s. This is still an issue, though, because all that paint is still there, and the paint flecks off, and it creates dust in the environment where kids can breathe it in. So in older homes that are dilapidated, you can still find all kinds of lead just in the environment. And then for some kids, they get this weird condition where they are interested in eating paint chips. And if they're eating lead paint chips, they're going to get severely poisoned with lead. And we know that this is the case. So the third source actually stems from the use of lead in leaded gasoline. So in the United States, starting in the 30s up through the 80s, gasoline had lead added to it. It was tetraethyl lead, and the reason why you added lead to gasoline is that it reduced the noise of the engines. It made them quieter. That was the only reason to add lead. Just made them quieter. Did nothing else to the engines other than make them quieter. So I mean that's a reason to add it, right? But the problem is that when you are then using the gasoline and you're burning off the gas inside the engine, there's exhaust, and the exhaust is now creating an aerosol of lead. And so now lead is all around in the atmosphere. And we're quite sure that it was leaded gasoline that ended up being a major source of lead contamination across the United States. So this is probably what's happening in these urban areas. So this is a picture of New Orleans. So this is a map of New Orleans, a paper from 2007 showing the concentration of lead in soils. And you can see that it is in the dense urban areas where you can still find really high levels of lead in soils. Now, the way we measure lead in soils is that we measure this by milligrams per kilogram of soil, okay? So I know I'm throwing out all kinds of different measurements of lead. In general, it's thought that anything greater than 400 milligrams per kilogram in soil, especially if children are coming into contact with it, that's considered to be unsafe, and that would require some kind of mitigation. Now, in urban areas, we know that these are the case, but no one does anything about it. The lead is still there, and no one does, it doesn't change anything. And you can see that in these dense urban areas, lead levels are much higher than 400. In fact, this is upwards of 1,000. And the highest measurement that they had in this study was 1,768. Really high levels, like more than 4 times the amount that's considered to be unsafe for children. The other thing that they did in this study is that they measured blood levels of individuals, and they compared those individuals to the soils where they lived. So these were children actually in these areas, and they found this very similar relationship to what we see with IQ score related to median blood lead levels, right? So with median blood lead levels on this axis and median soil levels here. So the soil levels greater than 400 milligrams per kilogram, you can see that this is associated with concentrations of lead in the blood that's around 5 micrograms per deciliter. That's that important cutoff. This is part of the reason why we consider 400 milligrams per kilogram to be an important cutoff. And you can see for these children, they're all above 5 micrograms per deciliter, especially as in the higher concentration areas. You're much more likely to have elevated blood lead levels, upwards of as much as 18 micrograms per deciliter, if you live in an area that has relatively high concentrations of lead in the soils. And you can see that there's this distinct relationship here. What this is showing is that in these areas of New Orleans, and they've shown this in other cities as well, that if children who are living in these areas where you're going to find a lot of lead in the soils, you're more likely to find elevated blood lead levels. Why now? Where did the lead come from that's in the soils here? This came from the fact that there was leaded gasoline that was used for 50 years. And this is decades later, and the lead is still there. I mean, lead is a heavy element, and it's just not going to go away. It's going to sit there, and it'll be breathed in by these kids. So it's this is probably what's happening in Cleveland, in Detroit, also probably in Flint, to be honest. This probably still is the major source for lead contamination for kids across the U.S., is breathing in lead from leaded soils. So there's I wanted to show some data about the historical changes in lead poisoning within the United States. This is a figure that was actually taken from Kevin Drum's blog on Mother Jones. He put this together. He was following the lead poisoning issues in Flint, and has talked a lot about the relationship of lead poisoning and crime, which we're going to get into in just a minute here. So this is the percent of children that had above 5 micrograms per deciliter of blood lead levels, or 10 micrograms per deciliter. So that's the red line. So this is giving us 2 measurements here. When Flint switched from the Flint River, they switched over to the Flint River, you can see that the percent of children that had 5 micrograms or 10 micrograms per deciliter across all of Flint went up. It went from probably around 2% of kids up to about 8% of kids. And then when they switched back to Detroit water, it went back down to around 2%, or like 4% basically. So using Flint River water certainly made a difference. But the crazy thing is when you look at the historical changes in blood lead levels. Oops. This is from Flint. In 2008, 2002, 1998, the percent of kids that had elevated blood lead levels was greater than 40% or 50%. This was not coming from the water source. This was actually coming from the fact that these kids were still being poisoned by lead from other environmental sources, likely from soils, and that coming from the fact that we used leaded gasoline for so long. So this is true in Flint, and this has been true across the United States, that blood lead levels have declined over the years quite a bit. And it used to be a huge, huge issue in the United States, much worse than it is today. So remember, so that's important to keep this in mind, that 5 micrograms per deciliter and 10 micrograms per deciliter is associated with an IQ decline. And what we're seeing there is not, we're seeing the percent of children that were exposed. So in 1998, you could expect that in Flint, around 50% of kids would have some effect on their IQ, and that a little bit less than 10% of kids would probably have somewhere on the orders of around 5% decline in IQ, right? 6%. Now, if you get higher than that, it gets worse and worse. Now I'm going to show you a data plot that shows the average lead levels in preschool children across the United States over time. That's what we're looking at here. Okay, so that's the green line. And this is showing the years. So starting from 1937 all the way up to 1988. This is the average, not the percent of kids, having above 5 micrograms per deciliter. The average child from the 60s all the way up into the 70s was somewhere between 15 and 22 micrograms per deciliter. That's the average. So this would mean that probably every single kid had greater than 5 micrograms per deciliter, in the late 60s and the early 70s. And so all these children are being exposed to lead. It's probably affecting their brains in quite a bit of ways. So, and then, of course, this is the timeline of lead reduction in the 1970s. The CDC said, 40 micrograms per deciliter is a problem. The EPA started to they started to phase out leaded gasoline. They banned residential lead paint. The CDC decided, if 40 is obviously too high, let's change it to 10 micrograms per deciliter, because we started to learn that the relationship to lead and IQ scores. And then it went all the way out to 2012 when the CDC said, maybe we need to consider 5 micrograms per deciliter as the level at which we can see some cognitive impairment. This is crazy to think about. So leaded gasoline started to be used in the late 30s, and you can see that the average blood lead levels went from less than 5 micrograms per deciliter all the way up to around 22 micrograms per deciliter in the middle of this era here. Now, one thing that's fascinating about this is that this change in blood lead levels seems to correlate pretty well with violent crimes. So the blue line is showing violent crimes per 100,000 people in the United States. And there was a crime wave that occurred starting in the late 70s and going all the way up through the 90s. So what this but then it started to decline in the early 90s across the United States. And it's continued to decline. In fact, violent crime nowadays is less than half of what it was in the early 90s. Early 90s was something else, let me tell you. But if you shift this by 21, 22 years, 23 years, something like that, you can find a pattern that seems to match violent crime pretty well. So if you were a toddler that was exposed to really high levels of lead early on in development, 20 some years later, you were more likely to be participating in violent crimes as well. That's what this suggests. This is just a correlation, clearly, but we know that lead exposure is associated with all kinds of behavioral deficits, not just IQ score, but increased incidence of ADHD, as well as violent behavior and impulsive behaviors. So and even in animals, this has been shown. So animals treated with lead, they have an increased violent response. So there definitely appears to be some sort of relationship here. And the late 20th century crime wave, some speculate, may have been due in part or on whole of the children having been exposed to really high levels of lead early on in development. And then when they got older, they just acted like little assholes and participated in violent crime and killed each other and did all kinds of things. And then things only started to get better once these people started to get a little older and they were less likely to participate in violent crime. And then for those children born in the late 70s and the early 80s, back now when the average blood lead levels were less than 5 micrograms per deciliter, they were much less likely to be participating in violent crime. So this is a bit of what I work on. How does lead affect the developing brain? So this is some of my more current work. And I'm not going to talk to you about it today. I just wanted to cover that relationship of lead and violent crime. And it's something I might revisit again and get into the literature a little bit more because I'm going to be writing a grant on this. And, I want to tie in some of those data because there's some other really interesting historical data on it that we might look at. So lead poisoning we know is associated with elevated rates of cell death in the developing brain. So this leads to eventually smaller brain size, it seems like. We know that it affects the expression of neurotrophic factors. So these are certain genes that turn on, that make proteins that are really important for supporting how brain areas connect to each other. We know that lead compromises dendritic outgrowth. So neurons have branches that have to connect to each other. Those are called dendrites. And lead can decrease how those branches grow. My particular area of interest is in thyroid hormone and brain development. And we know that lead affects thyroid hormone signaling. So that's really what my lab is focused on. And we're doing some interesting work on that I might talk about at some point when we get some of that published. So what is the mechanism by which lead impairs brain development? Well, that's for another day, but that's exactly what my lab is working on. So anyway, I hope that you enjoyed this. I'm going to be signing off here. I've been going on for almost 2 hours, so it's been a long time. So this is a long stream. I'm going to break this up into small segments. Yeah, I hope to gain an audience that will want to see these, and maybe we'll even get people joining on and chatting. I'm going to try to do this every Friday or Saturday. Yeah, so let's do it. Cool. All right. Everyone have a nice weekend, or, stay safe, and we will check you later.